Day 9, we're going to be continuing on with our lessons on the digestive system. This is week 4, and this is lesson 3, so you should be watching this on the Wednesday if you're following it in order. First off, because we record these a week before, last night for me, or yesterday rather, for me was when quite a lot of you took part in the Kahoot showdown that Miss Corbett arranged. So well done all of those people that went on to that and did that. Looks like it was a bit of fun. I think Miss Corbett will be arranging some more, so keep up the good work. It's just something a little bit different, and just keeps that motivation, which I know it's quite hard sometimes to have. So well done for all those people that took part. Well done for all those people that are continuing on with these lessons. Okay, so what we're going to be looking at today is catalysts, and it's going to be a type of catalyst which is an enzyme. Now, catalysts came up quite a lot in chemistry where you did rates of reaction, so you should be familiar with what a catalyst is. And we're going to be looking at a type of catalyst that we have in our bodies that we make, which are enzymes, and they're incredibly important in the digestive system. Okay, so before we get going, you'll be familiar with this because this came up on a previous lesson. I think it's the last lesson. We've got some pictures of some different parts of a balanced diet. So first of all, go through and identify those different parts of a balanced diet. And then for A, C, and D, can you have a go at saying what their function is? You don't need to write it down, just do it in your head, but you'll probably want to pause the video just to give you a minute to do that. Okay, so hopefully we remember these different parts of a balanced diet. We've got protein for A, and protein is really important. The function of that is to repair any damage to our bodies, replace cells that's needed to make new cells. Very important molecule, and also is the molecule that we're going to be looking at today, because enzymes are actually a protein, a type of protein. B is minerals. C, we've got carbohydrates, which are really important in getting our energy through respiration. You can see quite a lot of those are plant-based because they contain things like starch. D, we've got the lipids. So you've got the fat, like the butter that is shown, but also the liquid lipids, which are often referred to as oils. They're really important because they do give us energy, but also we store them and they're used to keep us warm. They're used on neurons to send messages around the body, and several other things. E is fibre. That's really important because that just pushes everything through the digestive system. Make sure that you've got a nice healthy digestive system and bowel movement. F is vitamins. So along with the minerals, these are really important to make the different molecules. So you need things like calcium for healthy bones and teeth and things like that. And G is water. Most of it is made of water, so it's quite an important part of a balanced diet. Okay, so we today are going to be looking at how enzymes work as biological catalysts. Now, bio means living, so biology is the study of living things. Biological means that they're made by organisms. So these are biological catalysts. They're things that we make that speed up a reaction. So the basics that we need to know is that enzymes are proteins. We need to know what happens with proteins. For 5 to 6s, we need to be able to describe how enzymes are used in digestion, and also talking about something called the lock and key theory that we will go into, which is very important idea theory of how enzymes actually work. And for 7 to 8, we need to explain how enzymes speed up reactions and how they control metabolism, which cropped up in a previous lesson. Okay, so enzymes work very similar to a lock and a key, hence the lock and key theory. Why is the shape of a key important? And can you think of other examples where shape is also important? So just take a few seconds to have a think about that. Don't need to write any of that. Okay, so why is the shape of a key important? Because it has a specific shape that will only fit specific locks. And that's a really important idea when we look at enzymes, because enzymes have a specific shape and they act like the lock. And then we have things called substrates, which fit in with those enzymes. And they're like the key. And it's very important that we keep using that term specific. They have a specific shape. Okay, don't write anything yet. Let me go through it first, and then you can pause the video and put down what you think is important. Okay. Now, we control a lot of reactions in our body. Hundreds of reactions are happening in our cells all of the time, and most of them are controlled by enzymes, so they can make them go faster, or in fact they can also slow them down. Okay, so catalysts are chemicals that can be used to speed up or slow down chemical reactions. A catalyst alters the rate of a chemical reaction, but remember, like in chemistry, it's not used in the reaction, not used up, in the reaction. So that means that you can use it over and over again. And enzymes, we call them biological catalysts. So the way that enzymes work is they speed up the reactions. They're made by living things, so they are biological catalysts. So you might want to pause the video now and put down what biological catalysts are. So enzymes are the biological catalysts and what they actually do. You only need a sentence, and the bottom paragraph is more important than the top one. That's just an example. Okay, so we use enzymes that are made by living things. We use these enzymes for loads of different things, not just inside our body, but we're going to be focusing mostly on inside our body. We use them in a process called fermentation. This comes up in another topic, but it's how they make beer and wine and the different alcohols. So we need enzymes in order to do that. We use it in bread making. And like with fermentation, we tend to use the organism yeast, and that makes our bread nice and fluffy and gets the alcohol in fermentation. We also use enzymes in yogurt production, so going from that milk, that very liquidy milk, into making things like yogurt. And also we can use it in biological washing powders. So most of the washing powders that we use to clean our clothes contain enzymes, and they break down all of the stains and things that we have on our clothes to clean them. But what we're going to mainly be focusing on is its use of enzymes in our body. Okay, and every single one of our cells is using these enzymes for hundreds of different reactions that are happening inside us. Okay, some nice pictures, just in case you didn't know what beer and bread look like. And that is a picture of a cell. Okay, so metabolic reactions. We spoke about this in a previous lesson. Remember, the metabolism is all of the reactions that are happening inside our body. It's not just respiration, but respiration is a good example of a reaction that's happening inside our body. And we use enzymes to control them, so speed them up or slow them down. Because we have got hundreds of different reactions happening inside our body, it's very important that enzymes are specific to only one type of reaction. If you had an enzyme that did lots of reactions, then it would cause havoc on all of the different reactions that are happening inside your body. You need specific enzymes that will only deal with specific reactions. So each different type of reaction is controlled by different specific enzymes, and that enables us to control the chemistry of the cell, those different reactions that are happening. Underneath you can see a computer model of an enzyme, and the different curls and lines and things like that is produced by that amino acid sequence. So we said about amino acids making proteins in the last lesson. Depending on the sequence of them, will give them these different shapes, very specific shapes. So no two enzymes, two types of enzymes will be the same shape. Now, enzymes are large proteins, and that's an important thing to remember. So protein in our diet is very important because it makes these enzymes. Because they're not used up, though, we don't need to make lots and lots of them. We only need to have a small amount that we can keep using. Now, there is a video link for this slide. So I'm going to go through this slide, and then I'm going to ask you to come off this video and then go to the show my homework, where I added the link that goes with this video, and it's just a very straightforward animation kind of summarizing that lock and key theory of how enzymes and substrates come together. But first of all, I'll go through what is happening on this slide. So we've said that enzymes are protein, and their specific shape is really, really important because it means that it will only bind or join to a substrate. And the substrate is what is the name that we refer to that fits into the enzyme. Now, as we've already said, chemical reactions in our body are controlled by enzymes, and they most of the time speed up the reactions. And like with catalysts in the chemistry topic, they do that by lowering the activation energy. Now, hopefully you remember the activation energy is the amount of energy needed to get a reaction going. So reactions will happen in their own time, but most of the time they take a very, very, very long time. So if we lower the activation energy of a reaction, it means we can get it going a lot quicker. And that's really important because if we eat a KFC Zinger Tower burger, that would break down naturally into the different parts, into the amino acids and the glucose and the glycerol and fatty acid chains, but it would take too long. We wouldn't be able to get enough nutrition from it from the amount of time it takes. So we need to use these enzymes to make it happen a lot quicker so that we're able to sustain our cells. You can see there is a picture under the video icon. There is a picture of an enzyme. The enzyme is the purple bit. The yellow bit is the substrate, and the bits on the right hand side are the products. So you have an enzyme that will join together with a substrate, and then a chemical reaction takes place and you form the products. Okay, now the specific shape of the enzyme where the substrate fits into it is called the active site. You do not need to draw that diagram because there is going to be another one that comes up later on that will have all of that in there, so you don't need to worry about that. But if you do need to take any notes from this slide, such as how they work by lowering the activation energy, and if you didn't remember what activation energy is, then maybe pop down the definition for that, the amount of energy needed to get a reaction going. But can you, after you've done that, go onto the video just to see a little animation of how it works, and then if you come back on to here. Okay, so hopefully we had a look at that little animation to see that it's what's the main thing it's getting across to you is that specific shaped substrates will fit with specific shaped enzymes. So an enzyme will only break down or build up, some enzymes build things up, it will only act on one specific substrate. And that's called the lock and key model, or the lock and key theory. So you can see on here, we've got a picture of an enzyme, which has got the letter E underneath it, obviously standing for enzyme. And then we have got a molecule, which has got S underneath it, and that is the substrate. So the enzyme and the substrate come together and they form something called an enzyme-substrate complex. So when they're joined together at that active site, that forms an enzyme-substrate complex. And when that happens, chemical reactions happen and the enzyme will form products, okay, which is what the P stands for. So as I say, it will either build things up, but most of the time they're to do with breaking things down. So can you, using a pencil, draw out the different pictures? Don't worry if it's not a work of art, as long as you've got the idea that the substrate is a specific shape to fit in with the enzyme. Can you draw the diagram of how an enzyme works using the lock and key model? And then there are 5 bullet points at the top. Can you pop those down in the appropriate place? So an enzyme binds a substrate in a region called the active site. Only certain substrates can fit the active site. Enzyme-substrate complex forms. Substrate reacts to form product. Product is released. So if we can get those down on your diagram. Pause the video now. Okay, so we're going to be looking at something now called optimum conditions. Now, when we did chemistry and rate of reaction, we knew that if we increase the temperature, the reaction will go faster, and that will carry on increasing. But with enzymes, there is a temperature where they work best, and that's the optimum condition. So if the temperature is too low, then there's not much enzyme activity actually happening, because they're not colliding with each other, not coming across each other very much because they're moving so slowly. It means that there's hardly any products being formed. So the lower the temperature, the slower the enzyme activity. Now, when you increase that temperature, you actually increase the enzyme activity. So the higher the temperature, the more the enzyme is working on those substrates. Okay, and it reaches something called an optimum, so an optimum temperature. Now, there's 2 conditions you need to know about. Temperature, which we're going to go through on this one. But there is another one called pH, or there is pH. And different enzymes have different optimum pHs. With temperature, though, it tends to be the same. Now, after you've hit the optimum, if you keep increasing the temperature, instead of it getting faster and faster and faster, it actually very quickly slows down and stops completely. And that's because the active site on the enzyme changes. So if it is too hot, the active site will change, and it means the enzyme doesn't work anymore. We say that the enzyme has been denatured. That is a very important word, denatured, because enzymes are not living things. They do not die, which many students write in their exam. They are denatured. And because they're denatured, obviously they're not going to work anymore, so the reaction slows down until it stops. So here is a graph that shows the rate of reaction against temperature for enzymes. And you can see that as the temperature increases, it gets faster and faster and faster till it reaches about 40 degrees Celsius, and that's usually the optimum temperature. And then if you go past that, those enzymes change, so they don't work anymore, they're denatured, and the rate of reaction rapidly decreases until it's not working at all. Now, it's got on here that most of the optimum temperatures are 40 degrees Celsius. Can anyone make a link about enzymes? Obviously, they're in our body, and the temperature of about 40 degrees Celsius. Anyone notice a link between those two? Okay, so maybe, I'm not expecting you to have made that link, but some of you might have realised that's about body temperature. So body temperature isn't just an accident that we will have a body temperature of around 37.5 degrees Celsius. It's because that's where enzymes work the best. Okay, so yes, after 40 degrees Celsius, stop working. So here is a picture that is just showing what happens when enzymes are denatured. So you've got the normal enzyme on the left-hand side, and if you have extremes of heat or extremes of pH, then you can see that the enzymes change shape, and obviously it's not going to fit with that substrate anymore, so it's not going to work. It has been denatured. Now, it's not a common pH for enzymes. Different enzymes work best at different pHs. So the ones in your stomach will have an optimum pH much lower because of the acid, will be a lot lower than the enzymes that work in the small intestine and in your mouth, which have a much higher pH, again, much more close to neutral. So underneath, it's just got a little sentence of what happens when enzymes are denatured. So you might want to, could you pause the video and pop down what denatured means? Okay, so we're going to move on to putting this all together. I'm not going to ask you to do both questions. You just need to pick one of the questions. So you can either answer number one, which is state what enzymes are and explain in detail how enzymes act to speed up reactions in your body. Or number two, explain how enzymes are important in the metabolism of a cell or organism. So can you pick one of those questions, please? They are 6 markers, so take your time in your writing. You need to have at least 6 points that are going to gain marks in this. Make sure that for number one, first of all, you have to state what enzymes are, but then you have to explain. So you're saying why that is happening. And then number two is just explaining. So you need to say how it is that they act in metabolism. So pause the video now, have a go at one of those questions, and then we'll go through some of the suggested answers. Okay, so hopefully you've had a go at that. If you grab a different coloured pen or pencil, and we'll go through some of the suggested points to see if you got 6 or close to 6. So if you answer question number one, the first part was stating what enzymes are. So you might have said that they are proteins. That would gain a mark. Something that's not on there, but something that would also gain a mark is you might say they are biological catalysts. Then you have to go into explaining how they work. So if you have said that the substrate fits into the active site, that would be a mark. If you have said that that acts like a lock and key, that would be a mark. If you've said that when the enzyme and the substrate join together, it makes an enzyme-substrate complex, that would be worth a mark. If you said that that speeds up the rate of the reaction, that would be a mark. Then if you said that it releases products, that would be a mark. And then if you said about the enzyme not being changed or altered, so it can go on and do other reactions or worse that effect, that would also be a mark. So give yourself a total of 6 on that. Now, for question number 2, I think this one was a little bit trickier. You had to explain how it was used in metabolism. So if you have said about how chemical reactions are really important in the body, perhaps you said about respiration releasing energy, that would be a mark. If you said that each reaction is controlled by a specific or different enzyme, that would be worth a mark. If you said that that means that lots of different reactions can take place at the same time, that would be worth a mark, or that they're not interfering with each other or having an effect on each other's reactions, then that would be a mark. If you said that enzymes enable cells to perform functions, basic reactions, that would be a mark. And then if you said that specific reactions carry out particular functions, so again with the respiration example, you might have said that carries out respiration, which releases energy, as an example, that would be a mark. We might have said about indigestion, about breaking down complex molecules like carbohydrates into simple ones like sugars, that would be a mark. And that's happening at the same time or simultaneously, that would also be worth a mark. So if you answered number 2, then toss up your marks, give yourself a mark out of 6. Okay, so the last thing that I'm going to ask you to do is kind of a little summary. We've got some different activities on here of the things that we have gone through. 1 to 8 is just filling in the blanks of the key information. Then it says about drawing a diagram to show how enzymes catalyze amino acids into proteins. So you're going to have to come up with a diagram to show an enzyme joining amino acids together to make a protein. So have a go at showing that. And then a diagram to show how they catalyze carbohydrates being broken down into glucose. So instead of building up like with the last one, this one you're taking a carbohydrate and you're actually breaking it down into glucose. And then have a go at some trickier questions, which are at the bottom, 1 and 2. Why is an enzyme specific for a particular substrate? And number 2, why can enzymes be used again and again? So slightly longer answers for those 2 questions. But if you could have a go at all of them. We will obviously go through the answers, so if you could pause now, copy and complete 1 to 8, draw your diagrams, and then answer in 4 sentences the challenge questions. Okay, so hopefully we've had a go at those. So question number 1, enzymes will speed up the rate of a chemical reaction. Number 2, enzymes are biological catalysts. Number 3, enzymes are specific because they only work on one substrate. Number 4, enzymes have a 3-dimensional structure. Number 5, the substrate fits into the active site of the surface of the enzyme. Number 6, with an increase in temperature, the rate of reaction increases. Number 7, enzymes can be reused, so only small amounts are needed. Number 8, enzymes lower the activation energy. So then hopefully you had a go at drawing a diagram. Everyone's is going to be slightly different, but hopefully we've got an enzyme that has got an active site that fits in with 2 amino acids. And then if you show those 2 amino acids joining together to make that protein. And then the next diagram, you'll have an enzyme that's got a specific shape that fits in with whatever shape you've done for carbohydrates. And then that forming the enzyme substrate complex, and then it being broken down into a couple of glucose molecules, which will be your products. We then have the 2 challenge questions. So number 1, why is an enzyme specific for a particular substrate? So you might have said, this is just a suggested answer, you might have put it slightly differently, but worst case of enzymes are large protein molecules. and the shape of the enzyme is vital for the function, so it's important for the function because it's got those amino acids that form that specific shape, and it's that specific shape that binds with a specific substrate. The important bit in that answer is going to be so they fit specific substrates. Number 2, why can enzymes be used again and again? If you've said about how they are not used up in the reaction because they are a catalyst or a biological catalyst, so they can be used over and over again, that will be brilliant. Okay, that is the end of this lesson on enzymes. There is a required practical for enzymes, which Miss Corbett will be going through on her video on Friday. And that's looking at optimum conditions of enzymes and what happens if you go to the extremes of that denaturing. But from this lesson, hopefully we will remember that enzymes are proteins, enzymes are biological catalysts. Hopefully we know that enzymes are used in digestion and other reactions, and they use something called the lock and key theory. And then for 7 to 8, hopefully we are able to say about how enzymes speed up reactions and that they're used to control metabolisms or the different reactions. And it says about the experimental catalyst, that's what you're going to be doing on Friday. So I hope you are all safe and well. Take care of yourselves. Keep up the good work. And I will be back for the next lesson, which will be tomorrow. Take care. Goodbye.